let's see, on Friday, we went over initiation of RNA transcription. How does the RNA know where to go? Why does the RNA polymerase know where to go so that it starts transcription at the right part, at the right point, right? And part of that has to do with the DNA sequence in terms of the Tata box, the promoter region, the area that directs gene expression. And then the other part has to do with proteins orientating the RNA polymerase at the right part, or setting it at the start site, okay? But once it gets put in the right place, it can proceed on its own. Does not need a primer. It will incorporate the RNA nucleotides, right, on itself and bring them in depending upon the complementarity rules for from the DNA template. Okay. Now, that was the first step of transcription initiation. The second step is elongation. All right. How does the RNA polymerase, once it gets started, continue to bring these RNA nucleotides in? All right. Basically, it untwists the helix, proceeds about 10 to 20 bases at a time. All right. And it progresses. This is what we call a processive enzyme. All right. Meaning it's processive in that it just goes. As long as there's nucleotides available and it's bound to the template, it will continue to go. Okay? About a rate of 40 nucleotides per second in eukaryotes. All right, a little less than prokaryotes because of the structure of the DNA. All right? And in this case, also a gene can be transcribed simultaneously by several RNA polymerases. Why is that important? You can make many copies of the mRNA at once. Right? Make many copies of the mRNA, which then can lead to lots of protein produced later on in the cytoplasm. Okay? So here's your RNA polymerase unwinding the DNA. Right? Here's your 5' to 3' end. Right? Here's your 5' end of the RNA, 3' end of the RNA. So it's adding new nucleotides here according to your base complementarity rules. You get the untwisting of the DNA template and production of an RNA transcript, of the primary transcript. Elongation's easy. It just goes as long as the DNA can open and nucleotides are available. How does a gene or how does an RNA polymerase know when the gene ends? All right? Mechanisms for this are different depending upon prokaryotes or bacteria versus eukaryotes. Okay? In bacteria, there is a terminator sequence. Okay? There's a sequence of DNA that actually determines this is the end. Okay? The RNA polymerase recognizes that and it shuts off. Okay? That mRNA then can be translated without any further modifications. All right, so bacteria, very simple. There's all the genes, whatever is transcribed is translated. Okay? Eukaryotes, there's a little more complexity. All right? RNA polymerase II, which is the one that makes mRNA, messenger RNA, transcribes what we call the polyadenylation signal sequence. All right? So there's a sequence of a run of A residues, right? That then allows a separation, right? That's the signal sequence where we're getting towards the end. All right? So it transcribes through that and then continues for 10 to 35 nucleotides, a little bit past, right, this polyadenylation sequence. The polyadenylation sequence is a run of adenines in the DNA. Okay? So it's going to have the RNA is going to have a run of uracil residues, okay, that will cause it to kind of lose its connectivity. Part of that is what happens between adenines and uracils. How many hydrogen bonds are there between those two? Those two hydrogen bonds, okay? As opposed to a guanine cytosine has 3, right? 2 hydrogen bonds, a little less of an attraction, and even uracil with adenine isn't the best connection. Okay, so if you have several of those, that loses the connection between the RNA and the DNA, that double-stranded hybrid, and that causes the RNA polymerase to start to lose its connectivity, its hold on the DNA template, and then that's why it continues for a little bit and then comes off. That's the termination sequence. So this polyadenylation sequence of DNA is now the signal for the RNA polymerase to stop. That's how transcription is terminated in eukaryotes. Okay, so from that now, before we get into editing, right now in a eukaryotic cell, we have this primary transcript, okay, which is 5' to 3'. Okay? Does that whole RNA get translated? No. Okay? In eukaryotes, there's a lot of junk DNA. All right? A lot. There's, on average, they say, estimate is about 2% of the genome of the DNA in your cells actually translates to protein. Okay? So that's a lot of other stuff that's there. Okay? There are parts of the genome that are not translated. We call this intronic. Okay? So this RNA sequence is actually made up of part that's protein, and then you have parts that are not going to lead to protein. Okay? So we call the parts of the RNA transcript that will code for proteins is called an exon. So in this case, this is exon 1, this is exon 2. Okay? And the stuff in between is an intron. We call this intron 1. And there could be more, right? More and more introns, however many exons, whatever it might be.
So I'm sorry, an exon is the part of the RNA that is used?
That is the part of the RNA that will code for a protein. All right, that the cell will recognize and will translate into a protein. All right? So, do we want to translate the introns if they don't really do anything? Is that efficient? No. Is it necessary? No. So, we have to, the cell removes introns and then connects exons so that the mRNA is going to be exon 1, exon 2. Okay, so this is going to take that part out. All right, the intron. This is what we call RNA splicing. We're going to splice together all the coding sequences of the mRNA from this primary transcript. Okay, so if in our primary transcript, here's our exon 1, here's exon 2, that's exon 3, right? We have to take out all the junk coding and then put all the coding sequences together. All right? That's RNA splicing. Okay? Splicing is one part of the processing of RNA. Okay? There's 2 other things that have to happen. We're going to go through splicing in detail. All right? Now, the other 2 things that have to happen is that we have to mark this mRNA so it can leave the nucleus. Okay? And with that, we're going to also provide a signal for where the ribosome is going to bind. Okay? So we're going to add things to this message. We're going to add something called a methyl cap, a 5' methyl cap. Right? So it's this group of, it's a methyl group, right? CH3 that we're going to add to the 5' end of the mRNA. That's going to allow it to leave the nucleus. All right? and it's also going to provide a site for where the ribosome is going to bind. Okay? And then at the end of the mRNA, we're going to add a run of A's, what we call a poly-A signal, poly-A tail. Okay? We're going to add a run of A's at the end so that we know that this RNA was made from us and not coming from a virus or bacteria that's infecting the cell. Okay? That's going to prevent enzymes within the cytoplasm from breaking this down. Okay? So 3 things have to occur in RNA processing. A 5' methyl cap, RNA splicing, and a poly-A tail. Okay? With those 3 things, now the RNA can move out of the nucleus and bind to ribosome to be translated. All right? Questions on that? So you see what it does. Here's processing. Both of the transcripts are altered. Usually some interior parts of the molecule are cut out and other parts spliced together. Splicing we'll talk about in a bit. Okay? We're going to splice exons together. Okay? So we have your 5' cap. It's a modified G protein. That's why I wrote it like this. MPG, methyl phosphate. That's a 5' methyl cap. The 3' gets your poly-A tail. Functions. Export, so that the mRNA can leave. It's going to protect the mRNA and also has the ribosome attachment site. Okay? Questions on the altering of it. Now we're going to get into, okay, so we're going to process this, and now we're going to, okay, so here's an example they use in your book. Okay, there's your 5-methyl cap. It's a guanine nucleotide with a couple phosphates attached to the 5' end. Now at the end there you see you have a 5' untranslated region, the start codon, stop codon. Okay, you have a 3' untranslated region, and here's your polyadenylation signal. Okay, that's your lots of the run of A's for the end of this. And then the poly-A tail that's added at the end. All right? So what you see in yellow is what's happening in RNA processing. Everything else is a result of transcription. Okay? Everything else in red is actually coded in the DNA. All right? The other stuff is added after transcription. So, splicing. All right? Most eukaryotic genes have non-coding DNA in them, or introns. Okay? So we have to splice the introns out and link the exons together. Okay? And you'll see that this is important for variation of protein function and structure. All right, so splicing. So here's an example of splicing. You have, in this case, 2 introns. You have 3 exons. We're going to splice the introns out, put the coding sequence together, and then you end up with your a 5' UTR, a 3' UTR, untranslated regions. They are still part of the mRNA, of the mature mRNA. Okay? But it is an untranslated region. It won't be translated into protein. Okay? That's your basics for splicing. Now, how does it do this? All right, there are proteins within the cell that will recognize ends of exons. Actually, they're gonna recognize the beginnings of introns. Junk DNA typically has a very conserved or similar characteristic sequence at the beginning of an intron. Right, so you know a boundary of where the intron's going to be. All right? And so proteins will attach to that and come together and produce what we call a spliceosome. All right? A complex of proteins that will remove the intron and link together the exons. okay? There's also a process of what we call alternative RNA splicing, all right? In the example I used, I had how many exons? The example I drew. I had 3, right? So exon 1, 2, and 3. Right? That codes for a particular protein A. Right? What if I spliced out exon 2 and the introns, and I linked exon 1 with exon 3? What would that do? Would that give you a different protein? Yes. Okay? So it gives you a different functional protein. This is what alternative splicing is, a way of removing and changing, removing parts of proteins and basically changing their function. Okay? The globin gene, we use this a lot. Hemoglobin, myoglobin, right? Globin gene is like 25 exons. Okay? And beta globin, which helps make up 2 functional units of hemoglobin, it comes from the globin gene, and it's a set of exons. And alpha globin, which makes up the other 2 subunits of hemoglobin, comes from the globin gene and has a different set of exons. Alternative splicing, because you link exons in different orders, okay, or in different sequence, gives you basically two separate but related proteins. Okay? And it may change function, it may delete function, whatever it might do. But it is a way of changing how that protein's made up. All right? All still coming from the same mRNA. This is the spliceosome. It's a complex of proteins and small RNAs that come together, link together, and what they call cut out the intron, what we call a lariat. It makes a lasso type structure with the DNA. There's an enzyme a ribozyme, you'll see what that is in a second. There's an enzymatic function that cuts that out and links the exons together. All right? The real process of this can be done, you should cover that in detail in genetics, if you decide to take it. All right? There's a ribozyme. Okay? So that enzymatic function that I talked about by cutting out the intron is actually done by a RNA, right? A RNA molecule that has enzymatic function. So we're talking about a non-protein molecule that's functioning as an enzyme. Okay, that is a ribozyme. Right? An RNA molecule that functions as an enzyme. And in this way, those small RNAs that make up the spliceosome, the introns basically, and those small RNAs catalyze their own splicing. So junk DNA will basically cut itself out of the coding sequence. Pretty cool, huh? That's okay. Questions on transcription? So at this point, we've produced a mature mRNA that is now possible for it to leave the nucleus, bind a ribosome, and be translated. It has been spliced. It has been added and altered at both the 5' and 3' end by adding a methyl cap and a poly A tail. All right? Questions? The next step is translation. Now we're going to take the code that's been transcribed in RNA, we're going to read it in 3 base pair letters and get our sequence of amino acids together. All right, so this is the flow of information from mRNA to protein through the process of parts of this, molecular components. We're going to need a ribosome. We're going to need the mRNA, okay? And we're also going to need something that's going to carry the amino acids into the ribosome. Okay? That is that carrying, that transfer of amino acids is a property of transfer RNA. all right, that will be bound and attached to an amino acid depending upon its three base pair anticodon, all right, a part of the tRNA that is complementary to the codon that's found on RNA. All right, that three base pair code. Okay? So transcription. So this is the overall process. Right, here's the codon. Right here. The codon is the three base pair word that is found in RNA, which then is translated by the anticodon on tRNA and corresponds to a specific amino acid. All right? In this case, you see the ribosome is bound in brown is the ribosome, and you have a growing polypeptide chain, all right, where amino acids are basically transferred from one tRNA to another, all right, and then the peptide chain continues to grow. So, structure of tRNA. It basically has what we call a cloverleaf structure. Something like that. You know, I'm not the best, right? But this part right here, so these areas are double-stranded RNA, right? This part right here is important. You'll have three base pairs that stick out. That is your anticodon. All right, that's the important to. This area up here is where you're going to get the attachment to an amino acid. Okay? So, your amino acid at one end and the anticodon, which is complementary to a codon on mRNA, is on the other end. It's basically about 80 nucleotides long. There's plenty of parts where it base pairs with itself. Right, it's basically like an L shape. You see what it looks like. It looks like a clover leaf. Here's your tRNA in a couple different representations. Right, there's your clover leaf structure. You get some double-stranded binding of parts of the tRNA, of the RNA sequence, and then your amino acid attachment site opposite the anticodon. Now, accurate translation. To make sure that the right amino acid is going to the correct, is being called upon by the correct codon, two things have to occur. Number one, the correct amino acid has to be attached to the right transfer RNA, right? So we want to make sure that if this is the anticodon for proline, we need to attach a proline to that amino acid, that tRNA, okay? And then second is that the anticodon-codon recognition, okay? The correct match between the tRNA and the mRNA. There is what's called the wobble effect, or the wobble hypothesis, is that sometimes with the base pairing between the anticodon on tRNA and the codon on RNA, the third base pair has some flexibility. Okay? And this is another example of the redundancy of the code that we talked about, whereby if the codon AAU is proline, as an example, and AAA is also proline, does it really matter if the anticodon is TTT or TTA? Not really, because they should be bringing in the same amino acid, right? So that's an example of flexibility. If there's only one anticodon or one codon that codes for a particular amino acid, that flexibility tends not to be present. Right, that flexibility is removed. Okay, so first we're going to have linking. How does a tRNA how is it linked to the correct amino acid? It's an enzymatic function, okay, where the protein enzyme, the enzyme is going to recognize both the amino acid and the correct tRNA. There has to be substrate specificity that fits in so that the amino acid phenylalanine doesn't fit into where the right enzyme that is going to fit a tRNA for alanine. Okay? So your enzyme here has to have the right substrates. So if this enzyme is going to be synthesize tyrosine tRNA synthetase, it's going to produce that tRNA, it is going to bind the right substrates. So how many tRNA synthetases do you think we need? If there's an enzyme for each tRNA, which then will also bring in the right amino acid, how many enzymes are we going to need? So 20, one for each amino acid. Okay? Other guesses? I would say there's more than 20. Because the specificity is not only for the amino acid, but also for the tRNA. Right? How many possible tRNA codons or anticodons are there that are used? 64 minus the 3, stop. So 61. We need at least 61 different enzymes because there's going to be, you're going to add whatever amino acid to the right tRNA. So depending upon the tRNA specificity. All right? So the enzyme is specific for both substrates. The tRNA and the amino acid is going to link to that tRNA. Okay? That ensures that the right amino acid is attached to the correct tRNA. That was kind of confusing. I think I worked it out. All right? Questions on the tRNA and what it does, the transfer function. Okay? The next part is the ribosome. Okay? Ribosomes are found in the cytoplasm or on the endoplasmic reticulum. They are a complex of protein and RNA. Okay? A ribosome basically comes in two parts: a large subunit and a small subunit. Right? They're based upon that. They're broken. One is what we call 18S. One is what we call 16S. 18 and 16S are sedimentary units. One is bigger, therefore it sediments when you centrifuge it in a solution to a certain part, and the 16S subunit sediments to another part. That's all. Okay, and that's how they determine the two subunits of the ribosome. Okay. The large and small subunit join to form your RNA complex, your ribosomal complex, with the help of ribosomal RNA. All right, so there's some RNA that's with that. And the large and small subunit basically clamp around the mRNA single strand. Okay? Parts of the ribosome. So here you see a space-filling model up top. You see a space-filling model of a ribosome, and the blue is the large subunit, the yellow is the small subunit. Within the ribosomal complex, there are three areas, what we call three sites: the E, the P, and the A site. E site is for exit, P is for peptide, A is for acceptor. All right? So the acceptor site is where the next tRNA is going to come in and bring the right amino acid. Okay? The P site is where the growing growing polypeptide chain is going to be found. And the exit site is where that used tRNA is going to leave the ribosome. Okay? So that's your site. This is the model right here. All right? What you see right here, if I can let it move. All right, here's the model. In this case, at any time, 2 of those sites, of the 3 sites on the ribosome, have to be occupied by a or tRNA. Okay? At any time, basically 2 of those are occupied. In this case, you have the P site, the growing polypeptide, and then the amino acid. The tRNA has gone off. It already ejected the other amino acid or the other tRNA that's been used. All right, that's empty. So now what's going to occur is that the amino acid is the growing polypeptide is going to go from the tRNA in the P site and be moved to the tRNA on the A site, and the ribosome will slide down. Right? So this is going to move this way. That one's going to move here. Right? It's just going to ratchet down, slide down, and now the E site is occupied, the P site is occupied, the A site is empty. Okay? And then the E site exited, and then a new tRNA will come into the A site. Here's your explanation of the 3 sites. And so, how do we build this polypeptide? Again, 3 steps: initiation, elongation, and termination. Okay? We already have an idea about termination, right? How do we stop translation? With a stop codon. All right, so we'll get to that in a second. You know that we need a stop codon. How do we initiate this? What's important for initiation, for starting translation? Start codon. We have to know where we're going to begin. Okay? But as part of the mRNA, there was also that 5' UTR, that 5' untranslated region, which is basically RNA that's upstream of the start codon. So what else do we need? We have to find a way to bind the ribosome, right? Yes? All right, so how do you think? What characteristic of the mRNA that left the nucleus could a ribosome use to bind the right end of the mRNA and start translation? Mike? The 5' methyl cap. All right? We specifically modified the mRNA so that it could exit the nucleus, and we gave it we determined which end was which. Right? 5' methyl cap and a poly A tail. So this is the front, and that's the back. Okay? So a ribosome needs to bind at the front, right? Not somewhere in the middle. Needs to bind at the front, so it's upstream of the start codon, and begins at the beginning of the mRNA. And the 5' methyl cap allows that to happen. All right? So part of the initiation is the small subunit binds to the 5' methyl cap, then the large subunit clamps over it, and then it will slide down. Elongation is the process of adding new tRNAs as they occupy the E, P, and A sites, and they slide down. Okay? All 3 stages require other what we call translation factors. Right? There are other proteins that are involved, not just, I'm just saying we're simplifying this to show that the ribosome is the main function. There are other factors that are involved. Okay? So your initiation stage, right, translation brings together your mRNA, a tRNA with the first amino acid. The first amino acid, how many start codons are there? 1. Right? That codes for methionine. Okay? The small subunit binds to the mRNA with an initiator tRNA that binds to the start codon. Then the small subunit moves along the mRNA until it reaches the start codon from there. All right, so you'll see what this is. Okay, so here's the start codon. It binds. That's your methionine, and then it clamps on top of that. The 5-methyl cap also helps to recruit this small subunit. That's the function of the 5-methyl cap. It will actually recruit this and then slide down until it finds the start codon. Then that methionine binds. Once the methionine tRNA binds, that then stimulates the large subunit to clamp on top. Now it's a complete process. That's initiation. What's open on the now clamped large subunit of the ribosome as it's clamped on the mRNA? What is now open? The A site is open. All right? So now the A site's open. A tRNA bound to the correct amino acid will come in and begin the process of elongation. All right? Start codon. Important because it establishes your reading frame. Right? What did we talk about reading frame? What do we say about reading frame? Sienna? They can't overlap. And we have to make sure the codons are read in the right order. Okay? Because if we change the reading frame, we may change what amino acids are being brought in. Okay? And there are other initiation factors that are also important in bringing the ribosome, kind of, like I said, oversimplifying this. Elongation. Amino acids are going to be added one by one to the previous, right? The previous amino acid at the C terminus. So we're going to link the N terminus, or the amine end, the amino terminus of one amino acid to the carboxy terminus of the preceding amino acid, making that peptide bond. Okay? Each addition involves elongation factors, again, oversimplifying. Codon recognition between the anticodon and the codon, make sure the right tRNA is there. Transferring or producing a peptide bond. And then the process of translocation, moving the ribosome down and changing where the tRNA is on what site it is occupying. Go from A to P to E. Okay? And translation 5' to 3' direction. This is the process of elongation. Okay? So we'll start at the beginning. We went through initiation, and here they've already shown the growing polypeptide. You could basically substitute that for the tRNA that's bound to methionine. Okay? The A site is open. The codon that's there binds with the anticodon on the correct tRNA, bringing in the right amino acid. Okay? There's a hydrolyzation of GTP, which then begins the transfer of peptide from the P site to the A site. Okay, you'll see actually right here. See this little red arrow? That's peptide bond formation. The peptide, the growing peptide, is going to be transferred from the P site to the A site. And as that occurs, so does the ribosome slide down. So you get the transfer, what you see here, and then the ribosome transfers down so that the peptide that's attached to the tRNA is occupying the P site. And then the used tRNA leaves. CN.
Why does it go from P to A back to P?
The peptide itself has to be kept in the P site. So it gets transferred. Really, this is showing it in a stepwise progression, but it really doesn't happen that way. So as the peptide bond is formed, so does the ribosome move down. So it's like simultaneous. Like this is showing it in a stepwise or simplified form, but it really happens at the same time. The peptide gets transferred from the the P site to the A site as the A is moving to the P.
So you just need the peptide bond in order for it to move down?
Yes.
Okay.
All right, we'll stop there. We'll pick up termination. We'll go over that again on Friday. We'll pick up termination of translation. Your quiz is everything from the last quiz up until today.
Can we make the quiz first?
I don't know.